Welcome to the wrap-up lecture in our series, Science Times. If you've enjoyed these lectures, you can thank tonight's speaker. When we got ready to put together this series honoring Erin Lemnick, members of the Adult Schools Lecture Committee met with Erin's friend and colleague, Shirley Tillman. And with her, we talked through what should be covered and who would be the best people to talk on each topic. The result is what you've been experiencing for the past several weeks. Occasionally there is a speaker who really needs no introduction. And probably in this town and to this audience, that's the case tonight. But I'm going to do it anyway. Shirley Tillman is a renowned scholar and teacher in the field of molecular biology and genetics. And of course, since June 2001, president of Princeton University. She was on the national committee that set the blueprint for the Human Genome Project, has done pioneering research in molecular biology, and is a national leader on behalf of women in science, and has led efforts to make the early careers of young scientists as interesting and productive as possible. All of that is the formal part, for the record, as it were. But I noticed that when Erin Lemnick introduced the then Professor Tillman to an adult school audience in 2000, he chose to focus on two contributions. The contribution to the university as chair of the President's Council on Science and Technology, which encourages the teaching of science and technology to students outside the sciences, and on her reputation as a truly outstanding teacher. If those were the two things Erin thought best described Shirley Tillman, that's good enough for me. And both of those attributes should stand us in good stead tonight, as she teaches this generally non-scientific audience about the science and politics of embryonic stem cell research. It's indeed a pleasure to introduce Shirley Tillman.
Thank you very much for that nice introduction. And I do remember Erin's introduction at the adult school over at the high school, which is where it was held in the past. And I remember saying as I was getting up to give the lecture, I think about the Genome Project was probably what I was talking about at that point, that it is very nice to come and talk to all my neighbors. And I still feel this tonight, having lived in Princeton for 20 years now. It really is a pleasure to be with all of you this evening, and I am greatly admiring of an audience prepared to come week in and week out to hear about science. You could not have known when you signed up for this lecture series that you were going to be treated to some of the most remarkable minds who combine not only distinguished science, but distinguished teaching and the ability to communicate. Individuals like my great colleague Bonnie Bassler, who I gather was a huge hit, and none of that, by the way, surprises me at all. I knew she would be a huge hit with all of you. I am going to, this evening, talk about embryonic stem cells, and I really have, I think, two qualifications to talk about stem cells. The first is that in my own scientific career, I have worked on embryonic stem cells going back to 1980, which is when we first started working on stem cells. Now, these were not human embryonic stem cells, which is why you haven't seen my name all over the headlines for that. They were, in fact, stem cells derived from mice. So that's one of the first sort of lessons of this lecture, is that we have been studying mammalian embryonic stem cells for 25 years. We know an enormous amount about them, but I'm going to also emphasize that there is still a lot we don't know about them. But that work did not elicit the kind of public attention that the discovery of comparable cells from human embryos, human embryonic stem cells, elicited in the late 1990s when they were simultaneously discovered by two independent groups here in the United States, one group in Wisconsin and the other group in Baltimore. And we're going to talk about why one obviously elicited so much more interest. My other qualification is really comes from a role that I played in the year 2000 and into 2001 when I was asked by the then director of the National Institutes of Health, Harold Varmus, to chair a committee that of a very diverse group of scientists, lay people, ethicists, lawyers, some public interest groups, to devise a set of ethical guidelines that the NIH could adopt as a way to regulate the study of human embryonic stem cells in the United States. Those guidelines were drawn up over a series of meetings that this committee I chaired had, and were delivered to Harold Varmus, who then delivered them to the Clinton White House, and there they languished for the rest of the Clinton presidency while he was otherwise engaged. And we're going to talk a little bit about what happened as we went from one administration to the next, what happened to those guidelines since then. So I've sort of seen both sides of this issue in my career, the scientific side as well as the legal and the ethical and the social policy side of stem cells. Hence the title of the lecture. So, we have to begin with definitions in science, of course, and that is what is a stem cell. And here I have shown you a stem cell that we understand better than any other stem cell in the mammalian organism, which are stem cells that exist in the blood, actually in the bone marrow. And these stem cells, these very, very primitive cells, give rise to literally all of the cells that make up the bloodstream. Everything from red cells to T cells and B cells, which are immune cells, to macrophages, which are again involved sort of in immunity, and so on. Platelets, which are involved in coagulation. All of these cells derive from a single stem cell. And these cells were discovered back in the 1950s. So we've actually been studying stem cells for a very long period of time. Now, what is the difference between a stem cell and a mature cell? And there are really three qualities that distinguish them. The first is that stem cells have the capacity to self-renew, meaning that when they divide, as these cells do very slowly in the bone marrow, they can divide and produce two cells that are virtually identical to themselves. And they can do that over and over and over again. And in fact, can do it to the extent that we consider these cells to be immortal. They could just keep doing this into eternity, renewing themselves. The other quality that is very important for a stem cell is that it has the capacity, once it decides not to renew itself anymore, but to become one of these mature cells, it has the capacity to become more than one kind of mature cell. And that's referred to as multipotent. And you can see that that is certainly true of the stem cells of the bone marrow. They become all these different kinds of cells, depending upon what the body's needs are. And the body essentially instructs the stem cell what kind of a cell to become. If you're anemic, they become red cells. If you are suffering an infection, they become immune cells. These are very smart cells in that they can be instructed by the body what to become. Now, mature cells, for the most part, have sort of the reverse of these properties. Not uniformly, but by and large, mature cells tend to become non-dividing cells. So they can't renew themselves. They can't reproduce themselves. And they certainly can't do so indefinitely, so they are mortal. Eventually, they will poop out. They will just divide so many times, and then they will stop dividing, and then they die. And finally, for the most part, and again, these are slightly, in everything in biology, you can find an exception. So I'm giving you the majority, not to suggest that something never one of these properties is not ever not true, is that they tend to be irreversibly determined. Once a red cell decides to be a red cell, it can change its mind and become a B cell. It's kind of made up its mind. So those are the properties of stem cells. Now, what is the ultimate stem cell? Well, the ultimate stem cell is the fertilized egg. It is the only totipotent cell. So toti means it has the capacity to become every single cell in the body. Because literally every cell in your body is derived from that initial fertilized egg that is a result of a male pronucleus from sperm, which is this little structure here, and the female pronucleus, which is this structure here, joining together, forming a single nucleus, at which point development proceeds. Now, the two-cell embryo, once that fertilized egg is divided, both of these cells are still totipotent. That's what twins are all about. You separate those two cells, each one implants in the uterus, and you get identical twins. So even at the two-cell stage, you have two cells that are still capable each of forming an embryo. And in fact, that's true through the 8-cell embryo. Every one of the cells here, this is divided twice again, can go on and form an entire human being, totipotent. But eventually, development begins to proceed as these cells divide twice again, and you form a structure here called a blastocyst. And I'm telling you about this because this is where embryonic stem cells come from. And in the blastocyst, there are two kinds of cells. There are the inner cells that are on the inside of what has now become a hollow ball, with inner cells that are the embryonic stem cells. That's where these are going to come from. But importantly, these cells are no longer totipotent. They're only multipotent. And the reason is they can't become one cell type. And that cell type is the outer cells of this structure, which are called trophectoderm. And they will go on to form the placenta. So embryonic stem cells are not like the fertilized egg. They can't alone form an embryo. So, how do you get these embryonic stem cells into a tissue culture dish so that you can now study them? Well, you start with, in my case, you started with some agouti mice. These are brown-skinned mice. You isolate fertilized eggs right after fertilization. Luckily, mice like to breed and do it with great regularity. And so it's pretty easy to get these fertilized eggs. And you can, in fact, grow them in tissue culture, or in fact let the mouse grow them for a couple of days, 3 or 4 days. And you can isolate these blastocysts with these two kinds of cells now, the inner cells and the outer cells. And if you just take these blastocysts and you literally put them in a petri dish with kind of good nutrients and things that they like to grow on, eventually the trophectoderm cells will die. Why? Because they are not stem cells. Right, they're mortal, they die. So we get rid of them. Nature gets rid of them. And all we have left are the stem cells, which continue to grow because they are immortal. Exactly. God, this is such a good one. All right. So we now have these embryonic stem cells in tissue culture. Now, how do we know that they are really multipotent? And how multipotent are they? How could you do a test to figure this out, what they can become? And in the case of mice, you can do a really elegant experiment, which we do all the time in the lab, which is we take embryonic stem cells into a needle, essentially, and we inject them into a blastocyst that we have isolated from white mice, albino mice. So inside the middle of this blastocyst are now going to be some cells that are derived from the tissue culture cells, the brown cells if you want, some from the blastocyst donor, the white mice. And if we implant this blastocyst now back into a mouse that has been fooled into thinking she's pregnant by injecting her with hormones, and that's called pseudo-pregnant, this will implant into the uterine wall, and you will get a mouse that looks like that. So you can see that the skin cells of this mouse are kind of a random mixture of the white cells and the brown cells. And then you can go through every single tissue in the body, every single one, and say, are there brown cells there? And there are kind of molecular tricks for being able to do it when you can't see color. And are there white cells? And sure enough, if you go through every organ of that animal, what you'll see is that the brown cells are there, and they are liver cells and nerve cells and kidney cells and pancreas cells, and you name it, you can find them. The only cells that couldn't be formed by these brown cells were the placenta that arose out of this blastocyst. And that's how, in fact, we know that those cells can't form placental cells. So this was the original test that told us that these cells have this tremendous potential to become everything in the body except placenta. Now, so that's an in vivo test, in life test. But what scientists want to do in the laboratory is essentially mimic what just happened in the body of that animal. And that is to take these cells in culture and find some condition, some hormone, some peptide, something, that if you added it to the medium, all the cells would become liver, or all the cells would become pancreas, and so on. That's the kind of holy grail of embryonic stem cell research, is trying to figure out what factor 1 through 4 is that would get these cells to do this. Now, can we do it today? The answer is no. We can do it for some cell types. For example, we're getting pretty good at making these make neurons in culture, nerve cells. We're very good at getting them to form bone marrow cells, and that's because we've been studying them for 50 years. We understand how to make bone marrow. But for the tissues that are listed here, we still do not have a clue. One of the messages of this lecture is how much we still don't know about how to use these stem cells to produce the kind of cells that we might want to use in order to conduct cell therapies. Because, of course, the goal here is to take cells that have been directed to one particular tissue and to allow them to grow up in tissue culture and eventually to be transplanted back into a human being who is suffering from some kind of disorder that it results from cells dying. And it's very important to understand that the only diseases for which stem cells are ever going to be useful are going to be diseases where the underlying pathology is the death of normal cells. And what you're going to be doing is replacing them with cells derived from these stem cells. So we're a long way from this, folks. Now, what are some of the scenarios where this might actually play out? I think the most likely at the moment is, other than bone marrow therapy, which we've been doing generations now for a variety of tumors, is juvenile diabetes. So juvenile diabetes is a complex disease that, as the name implies, obviously arises largely in children around the ages between about 5 and 15. That is the result of the death of the beta cells of the pancreas, which are the cells that produce insulin. And that's why children and adults with juvenile diabetes are completely dependent on exogenous insulin, injected insulin, which they need in order to metabolize glucose, which is the primary function of insulin, which is to get glucose into cells. So it's a disease where the problem is the death of cells. So you could imagine that if you could figure out a way to get stem cells to form beta cells of the pancreas and get those beta cells back into the pancreas and get them to settle in and to begin to grow and divide and not be killed by the body. Because remember, this is a body that has killed all its own beta cells. So you're going to have to figure out a way to make these cells so that the body doesn't attack them instantly. Then you have a potential cure for juvenile diabetes. It's a scenario that intellectually makes sense. It is a scenario that we are a long way from achieving. The other reason I'm very reasonably optimistic about juvenile diabetes is because some of the smartest people I know are working on exactly this problem. And that's absolutely key to having this work. Now, there are a number of liver diseases that are a result of the death of cells. Cirrhosis of the liver, for example, or extreme liver disease following hepatitis infection. One could potentially imagine that one could replace some of the liver cells that have died in those conditions with normal cells. So that seems also, at least to me, a plausible scenario for stem cell therapy. Parkinson's is a much more difficult case. In fact, I would say any disease that has to be treated or where this death of cells or neurons, as is the case with Parkinson's, dopaminergic neurons, it's going to be much more difficult. And the reason is that neurons function by talking to one another, by making connections from one cell to another cell. And if neurons don't make the right connections, it's like having a completely malfunctioning wiring diagram in any kind of a computer. It will just simply not work. And although we are very slowly learning a lot about how neurons normally connect up to one another during development, which is when most of it happens, sort of right before and after birth, we know nothing about how to get them to do it in an adult body. We just don't know how to do that. So when you hear people talking about curing Alzheimer's with stem cell therapy, don't believe it. Don't believe it. Don't fall for it. I think the likelihood of that is between none and none, frankly, given what we know today. No way. So, what stands in the way of stem cell therapy right now from a scientific perspective? We'll talk about legal and ethical issues in a minute. But from a scientific perspective, what stands in the way? And I think there are 4 major scientific hurdles that have to be overcome if we're going to really do this. First is the one I've already said. We still don't know how to get cells to choose one of those multi-potent paths of differentiation over another. And the last thing you want to do is inject a bunch of heart cells into a pancreas. Not a good idea. You want to inject pancreas cells into pancreas. So we're going to have to figure out what the rules of differentiation are. The second one is just a very practical problem. Here you have a juvenile diabetic who has a pancreas because there are lots of other cells in the pancreas that don't die. But how are you going to get the cells back in there? How are they going to set up housekeeping? What's going to allow them to set up connections to the blood supply so that they're getting oxygen, they're getting food, nutrients? This is about tissue architecture. And we know very little about how to engineer tissues in such a way that if by taking a big syringe and injecting those cells back into an organ, that those cells are going to home into the right place. A lot of research still needs to be done. This is, I think, a major challenge. And that is that cells are exquisitely sensitive to their environment. And one particular thing that they are very sensitive to is who their neighbors are and who is talking to them, both from near and from afar. And we know from a lot of work in mice that embryonic stem cells are not as stable as we would like as cells. If you grow them long enough in tissue culture, their chromosomes start rearranging, they start duplicating, and eventually they become virtually useless. And we know that the problem is the growth in culture, which doesn't mimic a blastocyst. We try to make it as much like a blastocyst as possible, but we don't do it perfectly. And therefore, these cells start acquiring mutations, acquiring chromosomal aberrations, and we know from a lot of work in mice that it's very easy to turn an embryonic stem cell into a tumor cell. Really easy. And one of the ways to do it is to put it in the wrong place in the body. So we need to understand this process much, much more before we're going to be, at least I would want somebody injecting these cells into my body. And lastly, we have a major problem, which is not a new problem. This is an old problem. It's been a problem since we've done organ transplantation in medicine, and that is unless the stem cells that are going to be used in therapy are derived from you or your identical twin, if you're lucky enough to have one, you're going to have to contend with the potential of immunological rejection. It's not an insuperable problem. And clearly, if we had hundreds or thousands of very good embryonic stem cell lines, we'd have a pretty good shot that there'd be one that would match up to you pretty well. Not perfectly. The only one that's going to match you perfectly is your own or your identical twins. So even when the match is pretty good, you're going to be under immunosuppressive therapy probably for the rest of your life. So that's always going to be an issue, no matter what. So, given some of those problems, there has been intense interest in finding alternative sources of stem cells. If embryonic stem cells have these problems, are there other kinds of stem cells that we could use? And indeed, we now know that most, not all, but most tissues in our body actually harbor a tiny number of stem cells. So if this is a normal adult tissue in this cartoon, you would have in the tissue a very small number of these stem cells, which are the red cells. And they would be immortal, because they're a stem cell, and they would be very slowly dividing. That's another quality of stem cells. They don't divide quickly, they divide quite slowly. And they give rise to, a slightly more differentiated cells, slightly more differentiated cells, and this is sort of meant to be sort of what would end up being a pretty complex group of cells. Everything from cells that are probably still dividing down to the most differentiated, the most mature cells, which would probably at this point be completely mortal and non-dividing. That's what kind of a normal tissue would look like. And these would be rare cells. I probably have drawn it slightly misrepresenting how rare they would be. In some tissues, we think they're on the order of 1 in a million cells. So they're not common, they're rare. Now, adult cells solve a number of the problems that we just went through with embryonic cells, but not completely. And they raise a whole new set of issues. So one thing it does that we'll get to in a few minutes is it eliminates the ethical objections to embryonic stem cells. Because if I can take a skin stem cell from you and figure out how to make it into a pancreas, no one, I think, would argue that there's an ethical issue with that. Here's the big advantage. It eliminates the immune rejection, which is what histoincompatibility means. Because it would be your cell we're injecting back into you. So your immune system is going to see it as self and you're going to be happy as a plant. So why isn't the whole scientific community rushing to these adult stem cells as the solution to the ethical issues surrounding embryonic stem cells? So the first thing to really emphasize is that we have just discovered these cells. We know almost nothing about them. If you figure that we've known about bone marrow stem cells for 50 years, these we've had for about 5 years. And there's a lot we need to understand about these cells before we even understand whether they are plausible as a replacement for embryonic stem cells. And the thing we really don't know about these cells is what is their developmental potential. So for example, if you take one of these cells out of muscle, what is its capacity? Can it become a liver? Can it become a bone marrow cell? Can it become a neuron? Or can it only become cartilage, bone, and myocytes, which are the cells of muscle? We're really at the dawn of trying to figure this out. So this is a field that I think has tremendous potential, and I'm quite excited about it. But it is 20 to 25 years away from our really understanding these cells well enough to know how they will do as a potential therapy. Now there is one very interesting thing that has come out of our discovery of these adult stem cells that I think is worth taking a quick digression to tell you about. And that is what these adult stem cells may be telling us about cancer. So here is the classical model for how tumors form in, and this is a model that I was taught as a graduate student, so it's, as my son would say, it goes back to the Pleistocene era. So here we have that normal tissue composed of a tiny number of stem cells and a lot of cells in the process of differentiation. And something happens, a mutation. Chemicals, X-rays, UV irradiation, whatever is the perturbation, one of these cells, and it could have been any of these cells for this purpose, one of these cells suffers not just one mutation, but a whole series of mutations, leading to a tumor cell. And that cell becomes a very rapidly growing population. And that is the tumor being formed right there with the asterisk in them. What is now being entertained, and I would say that this is new and this is still a lot of work needs to be done, is the notion that many tumors, maybe not all, but a number of them anyway, actually arise not by a mutation of one of these cells on their way to differentiation, but on the stem cell itself, and that the stem cell differentiating and dividing now more and more quickly results in the formation of the tumor. Now, why does it matter whether the mutation happens in a differentiated cell or in a stem cell? And the reason is because of what happens when you undergo chemotherapy. So here is sort of the rationale for chemotherapy in the classical model. The idea is that you give a chemical that is largely designed to kill rapidly dividing cells. So it kills these tumor cells, and it kills, under the best of circumstances, every one of those rapidly dividing tumor cells, and you eliminate the tumor. And that's really the goal of chemotherapy, although as all of us, I think, know in the audience, this goal is not always achieved. Now, what's the problem with this strategy if, in fact, many tumors arise from mutations in stem cells? Is that you have the stem cell, which is self-renewing all the time, and you have the tumor cells, and you give the chemotherapy, and it kills the rapidly dividing cells, all those progeny, the differentiated progeny, but it does not kill the stem cell because it's not rapidly divided. Remember what I told you, stem cells divide very slowly. And so what eventually happens is that the tumor grows back again. So one of the most exciting things that is happening in cancer biology today is people are beginning to think about how to target drugs to those stem cells, to those dividing stem cells, those slowly dividing stem cells. Because many recurrences as a result of chemotherapy may in fact occur, not because one of these cells were missed, but because this cell, by definition, will be missed with the chemotherapy. So I think this is one of the most exciting things that has come out of the discovery of adult stem cells. Now, let's go back to other sources of embryonic stem cells. And there is one other source that has now become infamous as a result of events in Korea over the last couple of years. And that is the creation of an embryonic stem cell, essentially artificially. So through the process of nuclear transplantation. And so here is the idea. You start with an adult cell, and it could be a fully differentiated cell, a mature cell. And you go in with a very fine needle, a pipette, and you pull out just the nucleus of that adult cell. And you inject that nucleus into an oocyte, not a fertilized egg, an oocyte that has never been fertilized, in which you've taken out the female pronucleus out of the oocyte. So the oocyte is just a shell, an empty shell, no genetic information. You put in the genetic information from this adult cell, and now what you have, in essence, done is you have assembled the equivalent of a fertilized egg. But the genetic information comes from an adult cell, not from sperm and egg. Now, under a very limiting set of circumstances and at very low frequency, that reconstituted cell will begin to think that it is a fertilized egg. And it will actually, at low frequency, develop into a blastocyst, out of which you can then take those inner cell mass cells here from the inside of the blastocyst, and you can make embryonic stem cells. So this is what Professor Wong in Korea claimed he had done, and claimed he had done it 14 or 15 times with 14 or 15 different adult cells from a variety of individuals. And as we all know from reading the New York Times, or probably even made the packet, it is now unclear to everyone involved in those experiments whether Professor Wong had really successfully done these experiments. They have been done, including in my own lab, for years with mice. So there is nothing technologically overwhelmingly difficult about doing this. This is a pretty standard procedure in experimental biology. But Professor Wong was the first person to claim that he had been able to achieve this goal with human cells. Now, why did Professor Wong want to do this in the first place? Well, it really comes back to immune rejection. What Professor Wong has done is had he done it, which looks like he didn't, but at least on paper he said he did, he has created a line of embryonic stem cells that would be genetically identical to the adult from which that skin cell or whatever the cell came from. And so you've eliminated this big immune rejection problem. So, what are the advantages of going through this process of nuclear transplantation? I've already told you the big one, which is that it eliminates histoincompatibility. Here's a debatable question, and the ethical philosophers among you can really go to town on this issue. And that is, there are some that argue that this potentially eliminates the ethical concerns surrounding the destruction of a human embryo. There are those who say that when you take an adult nucleus and you put it into a enucleated oocyte, you have not created a human being. You've created a sort of a reconstructed cell whose likelihood of going on to become a human being is very low, based on what we know about mice, which is about 1 in 300. Now, you could also argue on the other side of the case is that if you can do it 1 in 300 times, you've got an ethical problem. It has the potential to become a human being, and therefore it is no different than a fertilized egg. It is no different than a normally created embryo, and you've got identical ethical problems. So you can all fight about that, about which side of it of the argument you would take. This is a very serious problem that I think has not been seriously thought about by many in the field, which is a series of genes that I used to study when I had my lab, which are called imprinted genes. And the only thing you need to know about these genes is they are growth control genes. They are genes that regulate whether a cell divides or doesn't divide. And so these are the genes that you worry about with tumor formation. And we know, because we've done these experiments in mice over and over and over again, that when you go through nuclear implantation, these imprinted genes are completely misregulated. They've gone completely berserk. And one of the reasons why cloning, nuclear transplantation, trying to create an animal, is so inefficient is because these genes are really important for development. And no one has really figured out yet how to get these imprinted genes back under proper control. And we don't know a whole lot about what the potency of these cells are. Although in mice, they have the capacity to be very potent. But it still is completely unclear in humans because they haven't been made yet. So, do I think this is a potential for replacing embryonic stem cells? I would say the jury is still out on this. I'm not at all persuaded that this is going to be ultimately the way to go. So, what are the ethical issues with embryonic stem cells, with using embryonic stem cells? And I think we all know what they are. That in order to isolate those cells, you have to destroy the blastocyst. There's no two ways around that. The blastocysts in question, in the case of human embryonic stem cells, have, up until now, been created in the process of in vitro fertilization, which is a very common procedure now in this country. It's done in lots of clinics all over the country. And because of the inefficiency of IVF, it is a very common practice to superovulate the woman, create lots of blastocysts, because it would be ethically unacceptable to inject a lot of them, because then you're looking at quintuplets, which is an overabundance of joy. What happens in the clinic is that 2 or 3 are implanted in the uterus of the woman undergoing IVF, and the rest are frozen in liquid nitrogen freezers. And one of the big difficulties for these in vitro fertilization clinics is that they are building up huge stocks of these frozen embryos, and the question is, what is going to happen to these embryos? But these were the source of embryos that have been used to create the stem cell lines that currently exist. The other, as we just discussed, is at least the potential of creating cells through nuclear transplantation, although that is yet to be achieved. And we can debate whether that nuclear transplanted cell is the equivalent of an embryo or not. So, the ethical debate really comes down to these 2 issues. It comes down to whether in your set of beliefs, whether they are derived from religious beliefs or whether they are derived from philosophical positions, if you believe that the fertilized egg or the blastocyst has has the immoral equivalency of a human being, then you have to be opposed to the destruction of the blastocyst or the destruction of the embryo, because it would be the equivalent of murder. And those who are opposed to embryonic stem cell research use that word all the time. They see it as murder. For others, the issue comes down to weighing the potential benefits that come from stem cell therapy. And I've told you that I think those are not certain yet. We'll never know until we do the research, but I don't want to leave anybody with the impression that this is a guaranteed outcome. I think there's a long way to go before we're going to know that this really will work against the destruction of a human embryo. In other words, these people sort of view the ethical debate around the issue of whether curing disease versus protecting a life. How do you weigh those two goods in your own mind? And needless to say, these are issues that cannot be mandated, they cannot be legislated. These are ethical issues that each individual has to decide for themselves. Now, there's a second kind of ethical issue around embryonic stem cell, which really assumes that you're going to move forward with the research. In other words, moves forward with the notion that for some individuals, the destruction of an embryo in order to create stem cell lines is an ethically acceptable decision. And then there's a whole new set of ethical issues that arise in that case, and that is that the donation of those embryos have to be done in an ethically acceptable way. So what does that mean? It really means that the donors have to understand very clearly the purpose of the research, and I would say have to understand the uncertainty of the research. In addition, they have to be able to acknowledge that the embryo will be destroyed. It must be clear to them that that is going to be the outcome. It must be very clear that there will be no effect on their clinical care by refusing to donate the embryo. And in fact, the committee I chaired recommended that no couple should ever be approached to donate a frozen embryo until they had a successful IVF, so that there would be no ambiguity, no uncertainty about future care. That no embryos would ever be transferred to another uterus. That has to be a given, I think. That there would be no benefit for the donor anticipated. We do not want to get into the business of having this be products that get sold. And finally, that the source of funding and the expected commercial benefit need to be very clearly understood, if in fact it's in a circumstance where there will be a commercial benefit. So these are the kinds of conditions that we laid out. The other one I should say, which is a little bit, which is related to this one, no effect on clinical care by refusing, is that we recommended that the person who approaches the couple who has successfully undergone IVF cannot be the doctor who treated them. It has to be a completely independent person who is anonymous to them and to whom they feel no gratitude whatsoever. So you're trying to set up as neutral a situation as possible so that if a couple decides to donate their embryos, they are doing it with no sense of either gratitude or pressure being applied to them. As I told you, those guidelines were never issued. They really languished in the White House until the end of the Clinton administration, and then, as far as I could tell, they were shredded at the beginning of the Bush administration. And what I want to just say in the last couple of slides is that we are in, I think, right now, an extremely ambiguous position in this country with regard to the research with embryonic stem cells. And I'll try and give you a sense of how crazy the situation is right now. Number one, If I wanted tomorrow to isolate and use stem cells that were obtained from an aborted fetus, for example, abortions are legal in this country, I could do that. It's completely legal. And I could use federal funds in order to conduct the research using those stem cells. There is nothing today in law preventing a scientist from doing that. So once the embryo is implanted, it is legal in this country to destroy it and isolate stem cells from it. On the other hand, if the embryo has not implanted into a human uterus, it is illegal to isolate those stem cells or to work on them, if somebody else isolated them, using federal funds. That is just ethically indefensible, logically indefensible, right? The one is completely legal and perfectly acceptable, and the other one is preventive. Both involve the destruction of a human embryo. The more rigorous rules are the ones that haven't implanted versus the ones that have implanted. Now, the use of embryonic stem cells for research using federal funds, and the federal funds is important because 95% of all biomedical research in this country is conducted with federal funds. In fact, it may even be higher than that, maybe 99%. I haven't done the calculation recently. So by preventing the use of federal funds, you, for all intents and purposes, slow down at a minimum and shut down at a maximum research in this area. So the use of human ES cells for research using federal funds is permitted for embryonic stem cells that were generated before 9 p.m. on August the 9th, 2001, but is illegal if the stem cell lines were created after that time. There is an argument that the Bush administration has used to defend this policy, which is they don't want to do anything to increase the destruction of embryos in the future, and so they accept the lines existed prior to 2001, but they don't want to participate in generating any new ones. But it is put scientists who want to study these cells in a very difficult positions, because at the time this edict was announced by President Bush, it was thought that there were 60 to 70 lines available in the country. We now know it is now under 10. And my friends in the business tell me now that about 8 of those lines have so many intellectual property complications associated with them, because they were all created in companies, that for all intents and purposes you can't use them. So there are only now a handful of lines. And although I told you that stem cells are immortal, I lied. When you grow them in tissue culture, they are not immortal. They eventually just start pooping out. They lose chromosomes, they gain chromosomes, they get mutations, and eventually you have to replace them. And we're hitting a wall where we are going to have no cell lines left that can be studied using federal funds. And I think that day is coming soon. Now, here's a real irony. Right now, there are no restrictions on the generation or use of ES cells created by nuclear transplantation. So if someone in this country did what Dr. Wong did in Korea, they could do it successfully. There would be nothing to prevent them, nor would there be anything to prevent anybody from working on the cell lines created by that process. Nuclear transplantation is not covered by the ban. So we have this peculiar set of regulations in this field that bear little logic. That is certainly, if you were going to sit down and sort of set up a series of regulations or guidelines, these are not the ones I think any reasonable person would set up, whether you approve of ES cells or not approve of ES cells. This kind of complicated mixture means that I think we're in a very ambiguous place. And clearly, for some time, I thought I detected in Washington an eagerness to get beyond this and to regularize some of these regulations, or to rationalize these regulations. I'm no longer convinced that's going to be the case, and I think we're looking at 2.5 more years until the next election. And then, depending upon who the next administration is, we may or may not finally get to a place where there's clarity. There's both ethical and legal clarity about the use of stem cells. I will say that other countries have been able to find a path to the ethical use of stem cells. Probably the country that's the furthest ahead today is Great Britain, which have very, I think, clear regulations about using embryonic stem cells and generating them. And that country has very, very fine embryologists who are working pell-mell on these problems. So the work is going on outside the United States. It is going on inside the United States, largely with scientists who can obtain funding other than the government, from organizations like the Howard Hughes Medical Institute, for example. But it is very, very modest in quantity. As you know, New Jersey and California and Wisconsin have all at least taken steps to provide state funding for embryonic stem cell research. In no case has this had much impact, I will tell you. The California bond issue is tied up in legal wranglings that looks terminal, and in New Jersey we're broke. So despite the best of intentions, there's no money. So to be continued. I would like to stop here and thank you for your attention, and I would be happy to answer questions.
At one time, there was a lot of talk about umbilical cord cells. Are they viable for this purpose? And if so, how do they complicate the ethical argument?
Yeah. Their umbilical cord cells are the equivalent of bone marrow stem cells. There has been, unfortunately, very little research done on them to know what their potential is going to be. But I think
So the viability is still a question
it's
mark.
still very much a question mark. Yeah. Yes.
I have the same question, but following up. So they are actually differentiated cells?
They are cells that we think are pretty equivalent to bone marrow stem cells. So they're stem cells in the sense that they have all those properties of stem cells. But what we know about bone marrow stem cells is that their potential is to produce all those blood cells. We don't have, to my knowledge at least now, any evidence that they could become anything other than those blood cells. So they'll be good for that, and maybe in the future they'll be good for many other things, but right now we don't know. Yes?
What is the reason to believe that if you had stem cells that were able to do the function, liver, pancreas, or whatever, when you put them into a diseased organ, that there would be, the same disease would take those cells also?
Yeah. It's an extremely important question that you have asked. And the answer is going to be disease specific. So let me take the example that I'm the most optimistic about, which is juvenile diabetes. So juvenile diabetes is an autoimmune disease. And literally what happens is there are cells, immune cells, T and B cells in your body that literally attack the beta cells, only the beta cells, and destroy them. So your question is extremely astute, which is what's going to prevent the body from attacking the new stem cells? And the answer may be that we are now learning enough about autoimmunity that we could imagine engineering those stem cells so that they are invisible to the immune system. We're not there yet, but I think it is, I can imagine a pattern myself to having stem cells that are missing the cell surface receptors that the immune system is recognizing. Now, let's take the case of liver disease and hepatitis. If you have not cleared that hepatitis virus, then you're going to be in exactly the same state if you introduce new liver cells. The hepatitis virus, which is reproducing still in your liver, is going to find those tasty lunch just like they found the rest of your liver tasty lunch. So it's going to be very disease specific how you think about your very important question. Yes, in the back.
What strategies are being used to target chemotherapeutic agents to neoplastic stem cells?
Yeah, that's a really great question. One of the things we know about stem cells is that they have proteins on their surface that are unique to them. In fact, that's how we're finding them all, is we're finding that all stem cells have specific markers on their surface. Now, you can almost immediately see what the problem is. If you try and design, say, an antibody, antibodies are turning out to be fantastic chemotherapeutic drugs. That's what Herceptin is, for example. To that cell surface marker, is it going to eat up every single stem cell in your body? And if it does, you are cooked. Because the presumption is that those stem cells in your liver and your kidney are there for a reason. They're helping you rejuvenate those organs all the time. So what people are trying to do is sort of do a bifunctional kind of therapy where you're targeting the stem cell marker, but also a marker that might be unique to the stem cell in that organ. And so everyone's chasing around those kinds of markers to see if they can find them and try and figure out a way to take advantage of the unique profile of those kinds of cells.
And then you destroy all of the stem cells in that organ, correct?
Yes.
The healthy ones as well as the cancerous ones.
Yes, you do. That is the problem. And what you may have to count on is that the tumorous stem cells are dividing faster than the non-tumorous stem cells. That's what you may have to sort of count on. So it's tricky. It's tricky. Yes.
Is there not?
Oh, sorry. Then I'll go up. Yeah. No. Go ahead.
Is there not a misattribution of blame, if you will, on who is causing the death of the fertilized embryos? It seems to me that these fertilized embryos that are not used in IVF are essentially doomed from the moment that they were created in a quantity more than were needed, and that it's an illusion, and we're hiding behind it, to think that they're still alive and somehow useful. And 100 years from now, they're going to be thrown down the toilet.
What those who are opposed to the use of IVF embryos would say is that there are many infertile couples in this world who would love to have those embryos implanted in the female of the couple, and that and because there is always that option, anything other than that is immoral, unethical. And I would say that an argument that does not play well with that audience, which for a minute I thought that's where you were going, is, isn't it the right of the couple to make their own ethical decision about the disposition of their embryos? Why wouldn't we want to essentially allow that couple to decide for themselves? And of course, the response on the other side is, would we let everyone in the audience decide about murder? Is murder okay? Maybe you think murder's fine, and so we'll let you go out and murder somebody, but someone else doesn't think murder is appropriate. So you can see where you get. The individual decision-making doesn't work either. Yes?
You mentioned that work on adult stem cells is relatively new. What percentage of the work in this area would you say is on adult stem cells versus embryonic? And are there any federal funding limitations on adult stem cells?
No, there's no funding restrictions on adult stem cells at all, and lots of people are working on them. Very excited about them. I will say, before I came here tonight, I sort of wandered around the literature, the recent literature a little bit, just trying to get a flavor of whether people are feeling optimistic or pessimistic about adult stem cells right now. And I was actually quite surprised to see how pessimistic the papers are right now about the utility of these adult stem cells.
I actually have retinitis pigmentosa, and there's some work in California that they're doing with adult stem cells.
Yeah.
They've portrayed as very optimistic.
Yeah. You know, honestly, you can't be a scientist if you're not optimistic. It's just a fact of life. And so I'm an optimist, too. But I was surprised at what I sensed in some of the papers I quickly read over the last few days is how sobering the papers seem to be at the moment. Yes?
Could you give me any idea how long it will be before embryos or blastocysts that will become people will have a few stem cells removed and frozen with some regularity, so that as a child or as an adult they have an embryonic stem cell of their own?
Yeah, I don't know the answer to that question, but I think I just don't know. Technically, it's feasible. I mean, you saw that if you can put cells into a blastocyst, which is what we did with that chimeric mouse, you can take cells out too. Now, here's an interesting thought experiment, which I've been sort of playing with, along with others. I don't mean to suggest it's my original idea. If you went into a blastocyst and you took out 5 cells and you froze the rest of the blastocyst and you put the 5 cells into culture, you have not destroyed the embryo from which those blastocysts have come, for those ES cells have come. The blastocyst still goes back into purgatory, you know, for eternity in the liquid nitrogen freezer. Would that be ethically acceptable?
They do that presently today when they're testing for diseases. They pluck out a cell from the blastocyst, and the embryo is still can continue.
That's correct. That's correct. So I say I don't think
What do the ethicists say about that today? Do they agree to the concept?
I think they are not troubled by that, and I think one of the reasons they're not troubled by it is a single blastocyst cell cannot form an embryo because it can't form the placenta. And so it is not a totipotent cell like a fertilized egg or like the whole blastocyst. A single cell from inside the blastocyst does not have the potential to form a human being, and therefore does not deserve the moral protection that the blastocyst or the fertilized egg is given. So I don't think they worry about that. But it's an interesting, very interesting question. Yes.
Could I ask a technical question? What happens if you take the cell inside a blastocyst and put it in the sphere on the outside that will become the placenta? Does it learn from its neighbors not to become?
It's really a great question, and the experiment's been done. So the answer is that if you do the experiment before a very specific cell division, when you go from 16 to 32 cells, you can exchange them. You can take an inner cell and put it on the out, and it will become, in its environment, a trophectoderm cell. You can take an outer cell and you can put it inside, and it will become an inner cell mast cell. At the next cell division, absolutely not. The outer cells have been totally determined. They cannot become anything but trophectoderm, placenta, eventually, and the inner cells can no longer make trophectoderm. So it's a very orchestrated decision-making process that's determined completely by whether you randomly end up in the middle or on the outside, what your fate in life is as a cell in a blastocyst.
Can you tell me what a stem cell line is?
It just means it's one of those inner cell mast cells that's been put into a petri dish, and because it has this property of being immortal and being able to self-renew, it just divides and divides and divides and divides, and eventually it fills the whole surface of the petri dish. And you have to then, if you want to continue growing the cells, you have to divide them. And so you take half and you put them in one dish and half in another dish, and then they'll eventually fill those two dishes, and then you have to do it again and again, depending upon how many dishes you want. But if you keep doing this and you keep feeding them and you keep giving them new nutrients, they will just keep growing and growing and growing and growing it forever.
Can I misunderstand you to say that of the existing stem cell lines, that about 80% are contaminated?
No, actually 100% are contaminated. 100% are contaminated. So one of the things that was not appreciated, and it should have been, and it was stupid that it wasn't appreciated, is that the way we isolate embryonic stem cells in these petri dishes and keep them from differentiating wildly into all kinds of different things uncontrollably, is we grow them on mouse feeder cells. These are mouse fibroblasts, which are essentially like skin cells that have been inactivated. So the mouse cells aren't growing, but they act like a wonderful kind of carpet that the cells like to grow on. Now, what's wrong with that? Well, mouse cells have all kinds of endogenous viruses. So there isn't an human ES cell line that is legally usable in this country that could ever be injected into a human being, ever. Because you would never, ever, for safety reasons, want to use cells that have grown on mouse cells, ever. So one of the things people have been working on is how to grow these cells without these mouse feeder cells, because they are a no-no. What I said was that of the cells that are left, that are still usable, there are about a dozen. Most of them were generated in companies, and in order to use them, you have to tie up intellectual property rights for infinity using those cell lines. And universities like Princeton will not allow investigators to sign agreements like that. So the actual number that can be used is much smaller than the number that are still left. Yeah. Yes. Yes.
Well, given the current political climate, how are schools like Princeton being affected? Are you losing your best people, or are they fleeing to England?
In fact, a graduate student in the Wilson School is studying that question, actually, right now, Aaron Levine. And he just brought me a paper the other day that shows that there has been actually a movement of scientists to California in anticipation of the big bond issue, the $3 billion bond issue, and to England. So it is happening. Yes. Yes.
What's coming out of English studies?
Well, I think what is happening primarily in England, and I was just in Oxford in December meeting with probably, in fact, one of the world's most brilliant ES cell scientists, Liz Robertson. She thinks that the most important thing that has to happen right now in England, and where you can work on these cells, is to work on those 4 big questions that were on my original slide. How to get them to differentiate, how to develop methods to get them back in the right place, in the right architecture for the tissue. It's really deep fundamental things that people are working on who are really serious scientists. The serious scientists, the scientists I respect, are not injecting cells into people right now. Yes?
Is it legal to create new stem cell lines using private funds?
Yes, you can do it with private funds.
Is this going on?
It is going on. So one of the most impressive people in the field is a brilliant scientist named Doug Melton at Harvard. who is using funds from the Howard Hughes Medical Institute. But just to give you a sense of what he has to go through, Harvard had to set up a lab for him that was completely apart from his regular laboratory. No one is allowed to take so much as a pipette tip from his Harvard Biolab lab to his stem cell lab. There can be no people working in the stem cell lab who have ever accepted federal money at Harvard as a graduate student or as a postdoctoral fellow. He has to have a 24-hour guard on his laboratory in Cambridge. His family has been threatened with death threats because he works on embryonic stem cells. I mean, this is serious business, folks.
Well, who's the government using, like the FBI, or they have like a stem cell police?
I don't know the answer to that question. What kind of auditing is being done by the federal government to ensure that no federal dollars are being used in Doug's stem cell lab. But he is being meticulous about this because otherwise he's breaking the law. At Harvard, even at Harvard, they don't break the law.
I'll take one more question.
Yes.
Hi. I have a question. You said there was this sort of schizophrenia in the law, and it doesn't surprise me because I deal with law for a living. So that's not really a surprise. It's unfortunate. I'm wondering, has anybody worked on, we heard about it way back when, and I'm familiar with the problem of glaucoma, which is cell death, basically in the back of the eye, the optic nerve, and the nerve fiber bundle. Has there been any work on neurons on smaller problems like that, glaucoma, and some of the nerve damage that's sort of traumatic damage, spinal cord injuries, that sort of thing?
Well, there has been work on spinal cord injury. In fact, a fascinating book. Any of you who are interested in this, it's really a fascinating book by Jonathan Weiner, who wrote, won the Pulitzer Prize for The Beak and the Finch, the book about Peter and Rosemary Grant in ecology and evolutionary biology. This is, it's a fascinating story, and in this story, it's about a young man who develops, I think it's ALS. I think that's right, it's ALS. Which is a neuromuscular disease. And eventually somebody, believe it or not, in Philadelphia injects stem cells into his spinal cord. Has zero impact on his disease, which is hardly surprising, because if, in fact, it was ever going to work, those neurons would have to get into the right place in the spinal cord. They would have to send projections to the right muscles. There were other projections up to the brain, so that when you think you want to raise your arm, you don't raise your leg. And needless to say, it did not succeed. But it gives you a flavor, this book, and I wish I could remember, I've read it.
My Brother's Keeper.
What's it called? My Brother's Keeper. That's right, My Brother's Keeper. It's a fascinating sociological study of desperation. And I hope one of the things that I've conveyed tonight is that I think there is real promise in stem cell therapy in the long run, but we are a long way away from it today. And for scientists who hype it right now, I can only say shame on you, because the lesson of gene therapy, which was a hyper-hyped field for most of my career, has yet to deliver a single FDA-approved therapy. And we should have learned from that. So.